Hello, everyone. Today we are here to learn about the fourth macromolecule in AP biology, which is the nucleic acid. So we've already covered carbs and lipids, and that was followed by proteins. And our fourth, last one that we must learn about is the nucleic acid. So that includes DNA and RNA, and they are both ultimately genetic information. So let us get started. So the fundamental property of a nucleic acid is to store and transmit hereditary information that's passed on from generation to generation. So the information is coded into a nucleic acid, whether it be DNA or RNA, and it's passed on to future generations. The basic unit of inheritance is called a gene, and literally a gene is just a sequence of DNA that codes for a trait. Now, we will learn more about protein synthesis later, but ultimately what you do is you take that sequence of DNA and you code it into a protein, which will then determine some sort of trait in your body. And then lastly, just some more basics. Remember that genes are found on what are called chromosomes. And chromosomes are wrapped up DNA. They're usually around different types of proteins like histones. And they pretty much hold the nucleic acid together. In humans, there are typically 46 in human cells. 23 pairs. You inherit 23 from the mother and 23 from the father. So that brings us to 46. Nucleic acids, they are composed of 5 basic elements. You have your carbon, which makes it organic. You have oxygen, hydrogen, nitrogen, and phosphorus. And we'll go over where those are found in just a couple seconds. There's 2 main types. We have our RNA, which stands for ribonucleic acid, and you have your DNA, which is your deoxyribonucleic acid. Before we dive into the structure of nucleic acids, I wanted to talk a little bit about the history, or should I say herstory, of the discovery of the DNA molecule and what it looked like. There were a number of very important figures that were working on the discovery. One of them was Rosalind Franklin. She was a British scientist who was working in a lab with Maurice Wilkins, and she was using a device called X-ray diffraction to figure out what the DNA molecule looked like. Now, she took a very famous photo, photo 51, and it was given to another pair that you probably heard of, Watson and Crick, and that led to the double helix shaped discovery by Watson and Crick. Now, of course, we know there are issues. They never really gave her proper credit. And I'm going to show you a rap battle that kind of describes the story in a little bit. Now, once Watson and Crick received Rosalind Franklin's picture, it did help them kind of visualize and recognize that DNA was in a double helix shape, and from that they could create these DNA models, which they published in a journal and were given credit for kind of discovering that DNA was a double helix and how the bases went on the inside and the sugar phosphate was on the outside of the backbone. This led them to receive the Nobel Prize in 1962 for their discovery, along with Maurice Wilkins. However, Rosalind Franklin was not awarded the Nobel Prize because she had passed away, possibly from X-ray radiation cancer. The monomer of a nucleic acid is a nucleotide, and nucleotides make up both RNA and DNA. Now, there are some differences between RNA and DNA, and we'll dive into those, but each nucleotide will contain a phosphate group, which is PO4, so 1 phosphorus and 4 oxygens. It will contain a 5-carbon sugar, but those will differ with DNA and RNA. It contains a nitrogen base, and they're all linked together by covalent bonds. So that means the electrons are being shared between the 3 parties to make the nucleotide. So let's look at the phosphate group. Again, the phosphate is composed of 1 phosphorus and 4 oxygens, and it makes up the DNA or RNA backbone. So if you're looking at this image here, the sugar phosphate backbone is the thing in gray, and so phosphate is part of that gray backbone. And you're going to notice that phosphate and sugar alternate in the nucleic acids. The sugar part of the nucleic acid does differ between DNA and RNA. So in DNA, the sugar is still 5 carbons, so it's composed of 5 carbons, but the sugar is called deoxyribose. In RNA, the sugar is a 5-carbon sugar, but it is called ribose. And the only difference between the two, if you're looking at the picture on the screen, is that deoxyribose is lacking 1 oxygen on the carbon ring, whereas ribose in RNA contains an oxygen. So the reason why DNA is called deoxyribonucleic acid is because it is lacking 1 oxygen. These are the items that will bind to the nitrogen base that form the interior of the double helix, and it's also the backbone of the DNA and RNA, along with phosphate. So this is an up-close picture of a comparison between deoxyribose, which is in DNA on the left, and ribose, which is the sugar in RNA. So you can see as far as chemical makeup, they really are pretty similar. However, you will notice that on the second carbon of the carbon ring, that deoxyribose is lacking 1 oxygen. So therefore, we call it deoxyribose. On to the nitrogen bases. So if we're talking about DNA, the nucleic acid, there are 4 nitrogen bases that you should know. So we have adenine, thymine, guanine, and cytosine. You're going to notice on RNA, that's a little different. Chargaff's rules say that DNA likes to pair with each other, the nitrogen bases. So there are some very important DNA base pairing rules that you must follow, you must adhere to, or we have mutations. So the nitrogen base adenine will only complementary pair with the nitrogen base thymine, and cytosine will only complementary pair with guanine. A good way to remember this is a very simple phrase, apple tree. So A goes with T, and then car garage. So C goes with G, and that will help you remember the base pairing rules. And what does hold the nitrogen bases together in the interior of the DNA molecule? It is our lovely friend, the hydrogen bonds. So hydrogen bonds will form between the nitrogen bases, and they will keep the DNA molecule in shape. This is an up-close picture of the adenine nitrogen base binding with the thymine nitrogen base. First off, you can probably notice that adenine is composed of 2 rings, whereas thymine is only composed of 1 ring. Also, when adenine binds to thymine in the complementary pairing, they form a double hydrogen bond. So that means they have 2 bonds that are being held together with hydrogen. Compare that to cytosine and guanine and their hookup. Cytosine again is a 1-ring molecule, whereas guanine is 2 rings. And when they combine, they actually form a triple hydrogen bond. So that means they have 3 hydrogen bonds that are holding those nitrogen bases together in the interior of the DNA helix molecule. Now, RNA is a little bit different. So RNA has the nitrogen bases adenine, cytosine, guanine, and uracil. Notice that there is no thymine involved in RNA. Okay, so they still have 4 nitrogen bases. It's A, C, G, and U. There is no thymine. Now, looking at the ring structure of the nitrogen bases, you probably noticed that some of them were single ringed and others were double ringed. Well, the single ringed nitrogen bases are called the pyrimidines, and that involves the cytosine, thymine, and uracil. However, the double-ringed structures are called the purine nitrogen bases, and that is composed of guanine and adenine. Another thing that you may have noticed is that DNA is double-stranded. So it has two sugar phosphate backbones that go on the outside, and on the interior you have your nitrogen bases. RNA, on the other hand, is single-stranded. It only contains one sugar phosphate backbone, and the nitrogen bases kind of stick out from that molecule. Something that is also unique to DNA is that it is antiparallel. So I really love this image because you can see that DNA goes in one direction on one side and then it reverses on the other side. We will talk more about DNA's direction later, but if you notice, there are these numbers on the side. One side of DNA will go from 5' to 3', whereas the other side is 3' to 5'. And you're probably like, what do those numbers mean? The numbers are indicating the carbon on the outside of the sugar phosphate backbone. So we'll talk more about that, but each carbon in a ring of DNA is numbered. And so when you see the 5', it's referring to the 5' carbon. And when you see the 3', it's talking about the 3' carbon that is bonding with another molecule. When the sugar phosphate backbone is forming in either DNA or RNA, the sugar needs to bind to the phosphate. And when that happens, it makes what's called a phosphodiester bond or a phosphodiester linkage. So when the carbon of the sugar binds with the phosphate, we call that a phosphodiester linkage. All right, so that concludes our basics of nucleic acids for Unit 1, Chemicals of Life, and let's discuss.